Can you hear me? Okay. So, brain is a main control center of human body, and electroencephalography is a technology of continuous recording of voltage fluctuations emitted by brain. And brain-machine interface can provide intuitive control of robotic prosthetic legs, exoskeletons, and other assistive technologies. And BMI is a direct link between the human brain and external device. And inherently, this is a closed loop system that triggered by brain activity, and it reads and decodes neural data and sends sensory feedback to patient. Therein closing the loop. And in the system, both the human and the machine are active learners, such that machine learns how to decode EEG signal, human data, and human learn how to control external device. And by placing electrodes on the scalp, EEG recording capture the neural signatures associated with motor commands, providing a window into the user intent to initiate, modulate, or terminate movement. But one of the major challenges in the development of brain-machine interfaces is designed to an accurate and robust real-time neural decoder. EEG signals contain artifacts from muscle activity, eye movement, and environmental noise, which pose significant obstacle to accurate interpretation. And preprocessing play a crucial role on mitigating artifacts and noise to increase signal-to-noise ratio. And additionally, feature extraction methods are distilling meaningful features from preprocessed data. And this enables robust decoding of commands and facilitates seamless interaction with robotic device. And most EEG-based brain-machine interfaces consist of several steps, including data acquisition, using headset with electrodes, signal amplification, filtering, and then data processing, feature extraction, and decoding using machine learning. And here is one of the examples of brain-machine interfaces. And in this work, scientists decoded human walking using LSTM recurrent neural network. So they started with high-pass filtering the data, then downsampling and filtering with artifact subspace reconstruction algorithm. Then then they continued with independent component analysis, and then they did really wide, heavy hyperparameter selection for their model, and then they was able to classify right swing or left swing. With another example, we have these two classes, stand and gait. So they clean data with H infinity algorithm, then applied common special patterns to extract features. And in this case, they used linear discriminant analysis to perform classification. To sum up all the literature review process through these three fields, like signal processing, feature extraction, and classification, we can say that on the first stage with signal processing, mostly they do channel selection and artifacts removal and increasing signal-to-noise ratio. On the feature extraction, actually, scientists extract different frequency bands and extract feature from different frequency bands. So additionally, they perform dimensionality reduction and used to represent data in different domains. And on the classification, during classification, we noticed that the state of the art, mostly used classical machine learning algorithm, and in recent years we noticed use of deep learning models. But however, every strategy developed and tested their own neural decoding algorithm, and um, we saw a gap that we as a scientists need to, so there is a need for large-scale comparative analysis to find best combination, because scientists mostly stated that they used one or another signal processing algorithm without proper explanation why they preferred them. So the main research question sounds like, what is the best combination of signal processing, feature extraction, and machine learning algorithm for accurate neural decoding of leg movement during walking? And we kind of asking this research question in the context of implication for brain-machine interfaces for control robotic prosthetic legs, exoskeletons for patients with physical disabilities. And during the literature review, we actually looked through lots of algorithms. Scientists used, tried, tested many of them, but to compare, we selected most commonly used algorithms. And for signal processes, this is artifact subspace reconstruction and surface Laplacian filter. For feature extraction, it's common special patterns and independent component analysis. And we want to test all this signal processing and feature extraction with 4 different classifiers. Two of them is deep learning classifiers, like CNN, convolutional neural network, and long short-term memory, and 2 classical machine learning, like support vector machine and linear discriminant analysis. And to compare these combinations, we will use F1 score as a main metric. And also we will estimate some computational performance metric like training time and memory usage. And also to compare models, because the output of our research is a model. What is the best model and the combination of algorithms. So to compare such models, we will use Wilcoxon sign trunk test that is suited for such comparisons. So here is our data set. We selected a data set from open source. It was collected by neuroscientists on it was called Walk in the Park. And they did a pretty good job by labeling data, because they used 3D foot accelerometers to record leg movement. And this data set includes 15 young healthy subjects, and the data was sampled at 50 hertz. And therefore, in this data set, we're able to have classify 4 classes: right heel strike, left heel strike, right toe off, and left toe off. And to prepare our data set for semi-real-time conditions, when we have a short window of time, and in real time, actually, we need to be able to classify exact movement and at the exact time, what actually a person doing, so what is the gait phase. And that's why we had to divide our dataset into chunks with rolling window with 80% overlap. And the label of chunk is assigned by the last label of timestamp. Also, we use data from 22 electrodes. And in average, each training test is 13 minutes per subject, and test set for the test set we took last 2 minutes. So here I will explain in short some ideas of selected algorithms. So the first is artifact subspace reconstruction, is an automatic approach for noise removal, for artifacts rejection. So it consists in 3 steps with calibration, determining rejection criteria, and then rejecting noise components from each chunk of data. The second is surface Laplacian filter. It's estimate radial current flow at the scalp. And um, first of all, it's reference independent, and it preserves high special frequency activity of topologically close electrodes and attenuate low special activity. The interesting thing about this particular algorithm that it takes into account the 3D location of electrodes, and then it's able to highlight the useful signals in local area in each electrode. So here is a feature extraction algorithm we used, and this is independent component analysis. So it can separate EG signals into volume conducted activities of maximally independent brain sources and extract independent components. And these components actually serve as a features. And the last one is a common special patterns. This algorithm developed for EEG processing for classification, and originally it was developed for binary problems, but it can be adopted for multi-class classification. Okay. Here is a list of machine learning models for convolutional neural network. We selected EEG-NET. It's a widely used model in EEG processing. And we didn't do any hyperparameter selection. We used suggested hyperparameters for this model. The second is LSTM. And we also, this model was used in one of the state-of-the-arts I presented earlier. We also used recommended hyperparameters. But for linear discriminant analysis, we did hyperparameter selection, and also we did hyperparameter selection for support vector machine model. And also we selected radial base function for SVM, and it actually maps prediction onto a new higher dimensional space where they can separate classes linearly. And here is our experiment design. As I said earlier, we have 4 different algorithms, and all the combinations of them will consist in we have 9 combination of algorithms, and the base model is we build base model on normalized data. And in such setup, we have 36 unique combinations of EEG decoding algorithms per 15 subjects, and we have 540 total experiments. I had a question from a reviewer. He asked why do we have so much experiments, because particularly in EEG field in neuroscience, we each EEG signal from each brain signals from each person are different, and we have to treat those signals like different data sets because they're very different. And there is lots of explanation about this. That's why the common approach here in neuroscience, we have to build specific models per subject. But also there are researchers that try to build a general model. Okay, here is the results. And for convolutional neural network, on average, surface Laplacian filter and independent component analysis alone and combined resulted highest FN score. And also we did Wilcox statistical testing and compared models to our baseline. And actually here we compared this combination surface Laplacian with independent component analysis to our norm, like our baseline, and with significance level of 0.1, we received a p-value of 0.07, and with not such strict requirements, actually we can say that models are statistically significant. But if it take a significance level of 0.5, so we wouldn't say that. But anyway, this combination, surface Laplacian and independent component analysis, resulted higher F1 scores. For LSTM model, we noticed that actually artifact subspace reconstruction, artifact rejection algorithm play a good role, and we received the highest F1 score with this algorithm. But Wilcoxon test showed that the results are not significant, and there is kind of no statistical difference between baseline model and this model built on data preprocess with artifact subspace reconstruction. For the SVM, we found that actually surface Laplacian filter play a crucial role on brain-machine interface decoding, and we received most kind of highest F1 score with this preprocessing. And we can say that here feature extraction resulted low EEG decoding accuracy. And for LDA, in average, highest F1 score was achieved without additional processing other than normalization. And but for example, for artifact subspace reconstruction, we see that 6 of 15 subjects received high score. And also in some for surface Laplacian and independent component analysis, some subjects received also higher F1 score. But there is no kind of consistency in this results because actually EEG signals, as I said, very different from person to person. And some algorithms may work and some algorithms may not work. In summary, our research question was, what is the best combination? And in this setup, across all 15 subjects, many of the highest performing EEG neural decoders used with the convolutional neural network. But the method of signal processing didn't consistently have a significant effect across different classifiers and subjects. And actually, simple normalized data appeared to be slightly preferred. But on average, ECA feature extraction resulted the highest decoding accuracy. And also we measured performance metric like average training time per subject and model weight. And we can see that CNN stores only weights, and so the memory storage requirements are pretty low for CNN model. But and also for the training time, the longest training time was with LSTM classifier. Okay, and for the inference time, we noticed that, as I said, surface Laplacian filter kind of we received highest result with surface Laplacian filter, but the algorithm is itself pretty slow. But I think with we can speed up it with vector operations. And for the conclusion, so convolutional neural network resulted the highest decoding accuracy, outperforming all the other classifiers across every subject. And surface Laplacian filter and independent component analysis produced the highest F1 score with CNN classification. For the LSTM model, artifact subspace reconstruction resulted highest F1 score. And for SVM classification, we found that surface Laplacian gives us highest F1 score. And for LDA model, we found that it works better with just simply normalized data. However, ISR showed best results for 6 out of 15 subjects. But our work has its limitation. First of all, we tested our algorithms only on 15 subjects, and nevertheless, those subjects came from one data set, and it will be really interesting to see the results across different data sets. And also, this is a pseudo-online analysis. We'll try to make the inference really close to online, but anyway, it's pseudo-online. And this is, we did our research and experiments in open loop system. So we didn't use a robot, there wasn't a robot to control. And there is no sensory feedback and user training. Also, we had limited number of compared algorithms and limited hyperparameter tuning. Also, I can say that the fact of hyperparameter tuning produce kind of bias towards models, because I think, and according to the research, if we do hyperparameter selection for our deep learning models, we may achieve even higher results. And for the future direction, we would like to extend number of datasets and subjects. Also, we want to explore principles of transfer learning and develop a general model and compare the results to kind of personal models. And also, we want to increase the number of algorithms. For comparison, H-infinity algorithm widely used in EEG processing, and apply wavelet transform for also data cleaning. And for feature extraction, we would like to compare short-time Fourier transform and discrete Fourier transform, and test other classification algorithms like NeuroGPT, try to some new approaches, and deploy our decoding algorithms to evaluate our models in real time with exoskeleton. That's it. Thank you for your attention and time, and I'm really happy to hear your questions.
Thank you, Elena. I would really softly suggest not be really happy before hearing the questions.
Okay. It's stressful.
I was just looking at your thesis, and I've been lost in the contents, in the structure of your thesis.
Mm-hmm.
It says, introduction, chapter 2, methods, which are subdivided in data set, signal processing, feature extraction, machine learning. Then you go straightforwardly to results, where you have one small paragraph and 4 tables, as far as I do remember, and have the discussion. So the question comes, which you promised to be happy about. I wonder, where is the literature analysis here?
Literature analysis is here.
The methods.
It's before the methods.
Before in the introduction.
Yes.
Which is odd, as for me. Okay. Where is the problem setting? Which problem are you attacking in your thesis? No, I'm not looking into your presentation.
Okay.
I'm looking into your thesis.
I kind of explained what that.
Excuse me? I'm not looking into your presentation. I'm looking into your thesis.
Into my thesis. So
yes.
The problem setting is in introduction section. It's logically contain all this parts.
Where is the methodology used for achieving at the solution state?
So solution, what do you mean by experiments? Or solution?
This is the next question. What is the contribution of this thesis? The experiments, recommendations, and any new architecture? What is the contribution?
Okay. When scientists kind of have his kind of problem of neural decoding of gate, he needs to make a decision what algorithms to use. And what he can do, he can look through several papers, and what would he find? He finds that each scientist use its own brain-machine interface, and there is no kind of general recommendations according what, especially for for gate decoding, there is no recommendations what algorithm to use. Why scientists selected one or another algorithms? That's why we need to compare to make sure that we are using best possible brain-machine interface. We need to compare several algorithms, and some papers do very limited comparison. For example, they use several frequency bands, and that's it. And
Okay. So just please don't repeat what have been already said in the presentation. I would rather ask you, as far as I understand, in the context of this answer, your contribution is a series of cross-evaluation experiments to find the best combination of the
yes,
components
correct.
and give the recommendations for the future scientists,
Yes.
right? Okay. So it's rather an experimental study. Where is your experimental plan in the thesis?
I have experimental planning in thesis is the last paragraph of machine learning section
Excuse me?
where I explained. Yes.
But this is about the method. It's not about experiments. I'm asking, where is the experimental setup? Are you experimenting only with machine learning methods?
I'm experimented with signal processing, feature extraction,
Yes.
and
And machine learning. So structurally, it should be somehow separated, I believe. So results mean experimental results, yeah? For the experiments that have not been set.
we set experiments in the at the end of machine learning section, which includes all signal processing and feature extraction.
And all the discussion comes to the discussion chapter number 4, where
yes.
where you discuss what?
Where I discuss, first of all, I discuss my results and compare my results to the results of state of the art, at least kind of some directions and trends. Then I discuss our limitation and compare our limitation to limitations of other scientists' limitation or the papers. And then also discuss future work and also compare my future work with future work of competitors.
I'm also very sorry to mention I miss conclusions here in the thesis. You have conclusions in your presentation, but you miss conclusions in your thesis. Why so?
I guess the conclusions is kind of explained in all the
so they're
the
meshed up into
part.
they're meshed up into the discussion section. But
please.
They're somehow mixed with the discussion section. So I have to read through the discussion section and extract the conclusions in the form of recommendations for the future scientists.
Because structure of because the structure of so because the structure of this last chapter discussions and next steps, it's kind of it's a overall explanation. It's logically structured with my results, limitations, and future steps.
Okay, thank you.
As I understand, you kind of looked for one paragraph, yes, that that sum up.
I'm just wondering, how do you see the structure of your thesis, and if this view is logical? Thank you. You explained it. Then could you please go to the number of combinations, 36, okay, somewhere in your slides?
Yes, I have it.
Yeah, here. Now the question is, how do you come up with this number?
Okay.
What are the ingredients in your combinations?
So I have the answer.
Yeah, great. As expected.
Okay. So we have, you see, in the upper side of the scheme, we have numbers for each combination, where I have separate feature extraction, then
So you have 8 blocks in the upper part of this figure. You have 4 blocks in
yes,
and if
yes,
you multiply 8 by 4,
yes,
it will result in 32. It's 9.
9.
Okay, sorry. Sorry, I just missed one. Now it's clear. Excellent.
I asked this question lots of times by my supervisors. That's why I had to come up with this really clear scheme.
Oh, it wasn't really clear. See, if it was so clear, I wouldn't have asked that.
Okay, agree.
And perhaps last but not least, 15 subjects.
Yes.
Do you think the sample is statistically representative to make judgments and recommendations in your conclusions, which are also missing?
Okay. First of all, during literature review, we found that many many experiments actually took, for example, 4 subjects or 5 subjects. So, and in my case, 15 subjects in this field, it's kind of a lot. The second, so please, what next you ask? Sorry.
Is this set of subjects statistically representative? Does it really cover all possible cases statistically?
I see. What do you mean?
And my guess is the wrong answer.
It covers this 15 healthy subjects that from there, so the scientists collected the results from them in one laboratory with one environment step. Of course, it's really interesting to compare, to see what is the best combination across different data sets. And my results, we can say, valid for these 15 subjects.
Okay, I'll just add one more question to elaborate into the detail of the sample. What were the ages of the subjects?
Okay.
Were they females or males?
We have a description. So actually, all such data sets are that collected. They goes with a really good explanation about subjects, and I guess I didn't kind of go in such details about age. So I think I might have this information.
I would be happy about statistical mean and deviation.
Okay.
Just not to go into the individual details, into the personal information.
This paper that I kind of have in my slides contains all the required data, and they, I think, as I remember, half and half, half men, half female.
Thank you. I will go and read. Yeah. This is it.
Thank you, Elena, for interesting presentation. So let us start with the reminding of the problem setup. So when we have, so X is actually EEG, and what is Y? So what we try to predict by our models?
So we, okay. Shall I kind of go to the results tables?
Answer as you wish, but I'm not quite sure. So I understand that input data is EEG data, and what is Y data, output data?
Output data is predicted class, current movement of gate, current phase.
Okay, so the class of the movement.
Yes, class of the movement.
And then I understand what, and then back to the methods you used. So CNN. Generally, we use CNN for 2D data. And how you proceed here with 2D data?
When the data is recorded, it's so when we record EEG data from human, it recorded with lots of electrodes, and such kind of electrode setup can have up to 250 electrodes and even more. That's why when we kind of take a look at one short data sample, we will see all the, in my case, 25 electrodes. That's why the data for one time point represented by 25 kind of 25 values. And that's why it's very, this format of data very suitable for EEG, for convolutional neural network, because it's actually 2D data.
So you have let's say like this kind of chart, and there is a y-axis. It's represented by those electrodes,
Yes,
25,
electrodes, yes.
and x data is a time step. And the intersection, you have some signal value,
yes,
right?
Yes, correct.
But generally, when we use CNN for the image processing, the images, they have like square view, shape, right? So equal x and y. And here, obviously, it's rectangular shape. How you deal with this?
So it's we deal with it with kernel size, yes. Because so this model EEGNet performs convolution on each channel. Yes.
So what is what's the size of the kernel?
Here I kind of didn't go through the depth of EEGNet, of course. So as I said, I used kind of this model out of box. And now I can't say for sure what is the exact number, so exact size of kernel for this model.
And the size of the input image?
The size of input image is 100 per 25.
And 100, okay, let's see. And if you go to LSTM, it's more suitable for signal processing, and it try to reproduce the next token, let's say. So that is why we use it in NLP. But you actually try to predict the class, right?
Correct.
So how you switch from totally different setup of the problem from CNN to LSTM, how you interpret your problem for the LSTM setup.
So the last LSTM kind of value we treat as class.
So you have a sequence of tokens, and the last token is the class.
Yes.
Oh, that's really interesting. You decide, so it's your innovation, or you
no,
find it in some literature?
I found this is in the literature, and this how kind of this model is kind of used for classification in EEG.
And
that's why as I said earlier, that's why I labeled data in such way. When I applied rolling window, you see, and I kind of labeled the whole kind of chunk by the last label of timestamp, because I have originally each timestamp has its label.
And the reviewer asked you, or more precisely, give this comment. So why didn't you try to train your models on whole set of persons' data, but you trained on each person?
Yes, it's a specific of neuroscience. First of all, as I said, because we should treat each data from each person like separate data sets, because human brain signals are really chaotic, and they may not have similar patterns because people may think differently. Yes, in major cases, we all have the same brain kind of parts, but there is we can just train model on one EEG data on one person and then classify and apply this model for classification of data from other person. It doesn't work like that.
Then, okay, but back to the practical part.
Mm-hmm.
If we're thinking about application in real life,
Mm-hmm.
so you need then. So I bought the device,
Mm-hmm.
I put it on my head,
Mm-hmm.
and then I train on my data, right?
Yeah, correct.
But for so what is the practical application of this device? So I can imagine again this brain machine interface specifically used for, so for instance, for prosthesis design.
Mm-hmm. Mm-hmm. Yes.
And it means that people,
When
person without
without
legs. So for instance, how
yeah.
to train data for the person without legs.
Okay, I can explain. There is a kind of approach in brain-machine interfaces. They used the kind of approach with motor imagery. The thing is when people think about move, how he performs it move. Actually, brain we can measure the highlights in the part of the brain that actually responsible for performing this movement, and that's why brain machine interface use this motor imagery kind of approach when person think about movement, and we can actually train collect data set during person imagining one
I see.
or
Thank you.
other movement.
Yes, I also have a couple of questions. Let me start with simple one. You mentioned accuracy as a metric, but what about more representative metrics?
Okay, I yes, I said about accuracy, precision, recall, but actually those final tables are with F1 score, and it's balanced F1 score because it's I kind of there is a problem with multi-class classifications. That's why I use balanced F1 score.
Okay. The next, it seems like some randomness about the models, because for some models you optimize hyperparameters and for others you just missed this.
Yes, this is the limitation of our study because there it went from time time constraints and kind of efficiency of the experiments. Indeed, studies, state-of-the-art studies, they do heavy hyperparameter selection for each model, all of course for deep learning also. But in case of in this study, we have lots of experiments, lots of combination to compare, and to kind of do a proper to to do a proper hyperparameter selection. Actually, we need to look for hyperparameter for each combination. So we have, for example, 9 data set for convolutional neural network. So we need to have 9 rounds of hyperparameter selection.
You understand.
That's why
Time limitations, I understand.
yes.
So next question is about feature extraction. You said that you use some algorithm to make this, but you may miss, yes, exactly. You may be missing very interesting features which this algorithm just don't find. You mentioned that the data is very noisy and so on. So maybe it's just an assumption. Maybe this algorithm is not very good for this exact data.
Uh, for example, independent component analysis is really close to original party problem. Yes, when we kind of want to separate voices from different people, and the same kind of we can draw the analogy to brain when we have different sources inside the brain, and when we measure EEG signal around the scalp, several electrodes can measure the same signal, and that's why this particular independent component analysis is really helpful, and it's widely used in EEG signal processing.
Okay, and maybe the most interesting question, as for me, in like absent result, you mentioned that not written result, I mean, okay, but present result. You mentioned that. Can you open this slide or one of the last slides about models? Which model? Yes, previous one, and this one. You mentioned, if I understand correct, the simplest models gives you the best results.
Not exactly. The best results, I achieved best results with convolutional neural network.
But this is one of the simplest one compared with LSTM, for example.
So
by the structure, by the architecture, by idea, by training cycle, by any parameter you could account.
So you state that the convolutional neural network is as simple as, for example, as
I understand in this way. Okay, but I'm right that CNN gives you the best results. Okay, very interesting. Very interesting.
I can explain even.
Yes, this is a question.
Okay, I can explain. Even for convolutional neural network, so by its architecture, so convolutions place as a special filters for EEG data, and it performs its own kind of feature extraction, and this model actually see patterns in the data chunks, in 2D data chunks, and also we achieved highest results with good results with surface Laplacian filter for some subjects, and surface Laplacian filter actually with add its knowledge of 3D placement. So, and this knowledge, CNN model doesn't have, CNN model doesn't have the knowledge about 3D placement. And this particular surface Laplacian adds this knowledge and it's kind of its value for CNN model. And also, there is a common kind of sense that we don't need any signal processing and feature extraction. We need only convolutional neural networks that extract all the feature, and we don't need even kind of filters data. But my research actually shows that we can gain from signal processing.
So very cool. Maybe I could like explain that. I mean, the simplest because they are the oldest and mostly known and so on.
Okay.
Okay, and you don't need previous like feature extraction. It's very interesting. Okay, thank you very much.
Okay, I have one question. You commented on independent component analysis, but independent component analysis assumes independent sources. Yes. While during walking, you have clear periodicity. So there is probably no independence. You have periodic signals. And that's first part of the question. And then if we are interested in walking, then the classes also alternate periodically, so you have clear pattern. So why so complicated things if you are just interested in predicting these four simple classes states?
We kind of selected this time window for our to divide our time series into chunks, to preserve the ratio between the shortest event. For example, when toe of the floor, and it's pretty short event. And actually, this dataset was divided according to the smallest event, and that's why in this short temporal domain, you see the brain. So we have kind of two parts of brain, and movement of one part of head kind of fires the neurons from kind of opposite part of brain, and that's why when we walk and we kind of take a small snapshot of data during walking, we will see that if we go with right foot, we will see the highlights on the left side of the brain that is responsible for walking. And there is those, and where those patterns kind of appears. And actually, this electrode placement allows to collect those patterns. See, because every time we have different patterns.
Just a very quick question from my side. I'm just curious, was there, like, are your results somehow aligned with the other results reported in this domain? For example, you mentioned that there are works that focus specifically on one algorithm, say convolutional neural network. Does your results align with the ones that others report?
Okay, it's an interesting question, and it was really hard to find papers where they tested several approaches, but we found several, and as far as we can just compare numbers, because those experiments were done in different circumstances, and those are different people. So to compare directly the numbers, we had to take those data sets and do our test on their data. But actually, we found some trends. For example, usage of artifact subspace reconstruction with LSTM, we found comparable results, reported better accuracy. So we found that in some papers draw recommendations like applying surface Laplacian with independent component analysis, and we found this in our research, where we achieved best results with these two combinations. So we compared the trends, and we found that our results are comparable, or at least have the same kind of conclusions in the papers.
Yeah, thank you for presentation. Can you please go to the slide where you split it, the sequence, like time split, as I understand? Yeah, I think the next one. Okay, so as I understand, like you just sliced it and you predicted like not a sequence of classes, but you predicted like one class for each.
Yes, for each chunk.
For each chunk. Why have you decided to do so? It seems like if you can just predict a sequence of of classes, you will have like some more context, like additional context that can increase the quality of your prediction?
First of all, I had some consultations with specialists in this field, and it appeared that actually it's a common approach for EEG data processing. And as far as we don't do any kind of prediction of EEG, we are predicting class. And by dividing data like this, this chunk kind of contains information about previous movement. And then we ask the model, what do you expect the last timestamp to be? And it works pretty well, actually.
Okay, and to the next question. So you have stated that you use last 2 minutes of data.
Yes.
And as I understand, there should be like kind of some gap because you have overlapping chunks. So in order not to have like the last trained sample overlap with the first test sample.
Yes, I prevented this by, first of all, so I have, for example, I have a time series per person, this whole time series. And the first step, I divided my data into train and test. And when I did time windowing, I did it separately. So I did it on. So that's why I prevented the kind of data leak.
Okay. Yeah, but anyway, if as you stated that you were training like a separate model for each patient, I'm just curious, what is the tendency of training and validation metrics and losses? Is there like kind of point of where it starts to overfit, or maybe like it can improve?
So my advisor and I decided to limit the number of epochs to 50 for every model, and that's why kind of we just train model with 50 epochs. And yes, I look at the losses and on train and validation, and I just kind of saw that for for some data it was easier to kind of to train model, and for some not, because of the data differences.
But there was like some point where it started overfitted, or it can.
I did so before. Okay, before we decided to use 50 epochs, I kind of trained model with more epochs to 200s, and then when we look at these charts, we saw that around the 50 epochs, we found that the model has it's so it the model have good performance, and it's not overlearned, and that's why it was reasonable decision.
Okay, I got it. Can you please go to the slide with model architectures? I think let's start from CNN one.
Okay.
Yeah, so I think like there was already the question about the architecture, and also it seems a bit like unnatural to use this conv2d for such particular input, because like when you have this convolutional kernel to the kernel, you expect that it will like move upwards, downwards, like with the same logic as it moves like left, right. But if you have like upwards and downwards like channel like different EEG outputs of different EEG sensors, and from left to right you have time, so the logic of the axis is completely different. Can you explain why have you decided to use such approach?
I read about architectures with that applied 1D convolution. It's also kind of reasonable, and I think this is a similar approach. Actually, I didn't kind of go deeply into the architecture of EGNet, but apparently, as you said, that X axis has different information, kind of logic from Y axis. That's why they apply this not square convolutions.
Okay, but you have mentioned like 1D convolutions, and they seem like they looks like much more logical, because you can just stack your EEG sensors outputs like channel wise, and then you will treat it like separate channels, which looks logical. Why have you decided to abandon like this approach?
So when I did a literature search, and when I did experiment setup, I wanted to kind of select a convolutional neural network. But the next question was which. And according to literature review, I found that this particular implementation shows really good results for EG data. That's why I just kind of listened to giants and selected this architecture. And otherwise, I had to kind of come up with my own, but it will be kind of separate research.
Okay, I see. And I think like the last one, so the features that you have used, they're pretty interesting, but they kind of maybe unusual for signal domain. You have mentioned that in your future work you're going to try STFT, short-term Fourier transform, which is a baseline for like signal processing tasks. So I'm just curious, why have you decided to start from such like not widely used feature representations rather than from some widely used like wavelets or STFT?
I made my decision on frequency of appearance algorithms according to my kind of subsample of literature, which I did with no-ball algorithm, actually. And it just was frequencies that I received. Those algorithm was most commonly used. Yeah, of course, short time Fourier transform also were there, but I decided to start with this one because it's kind of most obvious, I think, and commonly.
Okay, thank you.
Thank you for your questions.
I think we are done with the defenses, and we need some time to do results consolidation and conflicts resolving. So how 15 minutes. So we will need 15 minutes to stay alone in this room.